In this module, we will look at the processes of science, the study of life, and the tools of biology. We're going to be looking at a vast array of topics. There's many aspects of biology. So let's start first with just what is it? What is biology? And biology is the study of life. It comes from the Greek word bios, which means life, and logi, which means the study of. There's many different subdisciplines: microbiology, biochemistry, and population biology, just to name some. And this course is designed to give a broad overview of biology. Let's talk about some of those subdisciplines. Microbiology, for example, concentrates on biological processes and structures that are too small for us to see with our own eyes. Biochemistry is looking at chemical processes that make life possible. And population biology deals with the dynamics of many life forms interacting in a community. And since biology is such a vast field of inquiry, most biologists do end up specializing in one of these subdisciplines. But before you can specialize or before you can focus on one of these subdisciplines, we do have to begin with a very wide and broad overview. That brings us to, what do scientists really do? Well, real scientists use many methods to investigate their area of interest, such as some of those subdisciplines. But all scientists, no matter what their subdiscipline or what their interest, draw conclusions based on the best evidence they have available to them at the time. So scientists gather evidence through observations and inferences. Scientists use the scientific method to explain certain phenomena and answer questions. So let's answer, what is evidence? It's definitely a collected body of data, generally from experiments and observations. But evidence isn't enough. In order to convince scientists of the accuracy of your conclusions as a scientist or biologist, your evidence must have been gained through completing multiple trials that produce similar results. Multiple trials, repeatable experiments, these are the keys to scientific discovery. And for that reason, scientists rely not only on evidence, but the collection and record of that evidence through a process called the scientific method. This is a system that scientists use to examine and explain their observations and inferences. Evidence, data, these are really important, but recording and having repeatable experiments that produce similar results are just as important, because that is what makes evidence or result credible. So let's talk about those steps in the scientific method. Observation and questioning is first. Scientists will observe something in their natural world and then ask why, or why is that way? Why does something look this way, smell this way, occur in this step or process? There's also a hypothesis, which is going to be a testable conclusion or statement about their observations and questions. There's a prediction, experiment, conclusion, and finally, retest and repetition at the end, because that's always important to make sure that the conclusion or the results are credible. So the steps in the scientific method are observation and questioning, hypothesis, prediction, experiment, conclusion, and then, of course, retest and repetition. That leads us to observation, because this is that first step of the hypothesis. And observations are gathered through your senses. It's something a scientist would notice in their natural world. A good example of an observation is noticing the creatures around a body of water, such as salamanders near a pond, and the fact that they have curved tails, not straight tails. And then a scientist would ask questions about those observations. What caused this? Why do they have curved tails instead of straight tails? And these questions generally involve making inferences. And inferences are logical interpretations based on prior knowledge, experience, or evidence. Another example of an inference would be, for example, if you saw 5 bears in a river and then you inferred they must be fishing. You would have to make this inference based on prior knowledge you had, that bears eat fish, that fish live in rivers. So using prior knowledge, experience, or evidence, and then putting it together to make a logical conclusion or interpretation, that's an inference, which is a lot different from a basic observation. The next step in the scientific method is a hypothesis, and this is just a suggested solution to a problem. Remember, it has to be testable because we're leading up to an experiment. A good template is an if-then statement. If this, then then. And what a hypothesis does is predict an outcome. So a hypothesis should always explain what you expect to happen. It has to predict an outcome. It also needs to be clear and understandable. Very testable, again, we're leading up to an experiment, and it must be able to produce data, something that is measurable. And it is going to contain an independent and dependent variable. And we are going to talk more on these topics in a later slide, but a hypothesis is always going to test one variable or one thing. The thing being tested or manipulated by the experimenter is the variable that is being tested. And we're always going to call that tested or manipulated variable the independent variable. Let's look at some examples of hypotheses. Factor A has no effect on B. Factor A affects B. Or if we decrease the amount of light during studying, then the participant's study performance will decrease. How about chewing gum concurrently during studying, then during taking an exam, does not affect test scores? Notice how all of these predict or make a statement, or maybe even use inferences. Let's look at, again, the third one down. If we decrease the amount of light during studying, then the participant's study performance will decrease. You can probably think of the logical steps that an experimenter had to make before they wrote out this clear, testable hypothesis. They may have noticed that they themselves study better when there is more light. They may have noticed that they study in a dark space, that their test results are not as good, meaning their study performance must not have been as good either. So these are all kind of qualitative or sometimes maybe even quantitative observations that an experimenter made and asked questions about. Maybe ask themselves, why do I not do so well when I study in the dark? And this may seem like a very obvious answer. You can't see the test materials well. Dark, dimly lit areas might make you tired, less focused, less aware, less awake. But regardless of what led the experimenter to this hypothesis, notice that it's clear, testable, and it predicts an outcome. Whether or not their outcome will be proven true with the experiment they design has yet to be seen. Let's look at one other example of a hypothesis. So, the coloration of birds matches their environment so that they can hide from predators. That's the clear, testable statement. And the inferences that may have been drawn were that a lot of birds match their environment. And a lot of camouflage is in order to hide from predators. So if you put two and two together or make an inference, we can produce this predictable, testable hypothesis. And that brings us to prediction. Hypotheses do predict an outcome. They predict the expected outcome that scientists expect to see when they devise an experiment and test their hypotheses. Hypotheses are not always true. Hypotheses are not always proven correct, but they do always make a prediction. That brings us to usually everyone's favorite part, an experiment, which is simply a procedure to test the hypotheses. Experiments test one variable, and a variable is a factor in the experiment that is being tested or changed during the duration of that experiment. And remember, for a valid experiment, we must only test one variable, and the variable being tested, or you can think of it as being manipulated, that is always the independent variable. So we've been talking about variables. Independent variable has come up a bunch. We haven't quite mentioned control variable, but they can get confusing. So let's talk about what they are. We're going to look at a control, independent, and dependent variable. So an experimenter changes one factor and observes or measures what happens. That's essentially what happens in an experiment. So a scientific experiment follows those rules. The factor being changed or manipulated by the experimenter, that's going to be the independent variable. And the factors that are going to be produced because of that one tested independent variable, basically what's being observed or measured as a result of manipulating the independent variable, that's the dependent variable. So let's put it in a couple of different terms. The factor that is changed, that's the independent variable. The factor that is measured or observed, that's the dependent variable. So manipulated variable is independent. The responding variable in the experiment is dependent. So the dependent variable is dependent on the independent variable. But what about the control variable? Well, that's what the experimenter is making a special effort to keep constant. You want to keep other factors constant, other variables constant, so that we really truly are only testing one variable, which is the independent variable. So everything else that the experimenter is controlling for example, if you're doing a test with plants, maybe the amount of sunlight or the amount of water they get, as long as that's not what you're measuring, that would be controlled, so that doesn't end up affecting the outcome. So those factors controlled in order to make sure that an experiment is having one true independent variable tested are the control variables. But what's the purpose? Why do we care? Why do we need a control? Well, controls aren't being tested, but they're great for comparison. So control variables are used for comparison. And it needs to be noted that not all experiments are designed in a way that they allow for controls. But experimenters do try to control all variables that are within their power to control. So let's look at some examples. Let's see, if you were trying to test if watering plants with coffee causes the plants to grow faster than plants watered with water, you would need two groups of plants. So you have one group of plants you water normally with water. This would be the control group. And then you're going to have a group of plants you water with coffee. That's the experimental group. And you want the experimental group to be what contains the independent variable. And of course, that independent variable in this instance that you're testing is coffee. Is the fact that you're using not the traditional liquid that's given to plants, water. That's why your control group's using it to make sure that nothing else is going on in a similar environment. That is making your experiment go wrong. Instead, only your experimental group is using the manipulated or altered variable of coffee. Notice both groups are basically identical, or we haven't said anything else is different between these groups. So what you'd want to do is make sure between the group that you water normally with water and the group that you water with coffee is that we use the same type of plant in both, same type of soil. They're in the same environment, so their temperature, the amount of sunlight, all of that is kept constant. So the only difference would be what's used for watering. And that means that the control group is going to be a great comparison to the experimental group to see if coffee is really going to help stimulate the plants to grow taller, bigger, wider, however the measurements are taken, compared to normally just watering with water. Data would then be collected for both groups, and that's where the comparison would take place. Let's try another example. Suppose you want to figure out the fastest route to walk home from school. So you take several different routes, and you're going to time how long it takes you to get home by each route. You are going to be the only one walking these routes, And that's because you want to make sure this is a fair experiment. And since you are only interested in finding a route that is fastest for you, it makes sense that you would do the walking yourself. So the control there was that you were the one doing the walking, and you would have maintained a similar pace for each one. That way you could compare all the routes. So varying the route would be the independent variable. That's what you decided on. You, as the experimenter or the biologist, manipulated or decided you would test routes. So the tested variable there was the route, which route was being used. So that is the independent variable. The time it takes is a dependent variable because depending on the route you took, the time was different. And keeping the same walker again allowed for comparison of all these different routes. So making sure it was you and you walked at a similar pace, that was the control variable. And just one more thing, it is best, remember, to have a repeatable experiment. So you want to run several trials and you want to test an independent variable each time. You want to keep that independent variable the same. That way you have trials that were repeated and hopefully with similar results. That will give credibility to your data. So let's talk about valid experiments. Let's talk about that credibility. What's required for an experiment to be valid? Well, two groups usually. Very rarely can we not design an experiment that has a comparison. It does happen, but it is best to have two groups, a control and an experimental group. There should be only one variable different between the two groups, so everything else should be controlled for or contained so that the control and experimental groups can be compared. Let's talk about the data you would collect from these control and experimental groups. Data is just the results of the experiment, but it could be quantitative or qualitative. And quantitative data is just observations or any data involving numbers, anything that involves counting, measuring. A lot of times this involves the use of utensils or different equipment in a lab. Qualitative data is observations that have to do with things that aren't as easily measured. The color of something, the texture of something, identifying an organism as a bear or a salamander. Not necessarily something that would be measured, unless there's 5 salamanders, then that would be quantitative as opposed to qualitative. It's really important that your data be organized. It's best to write in straight lines, to create charts for yourself, and to review your data once you've written your chart or drawn your bar graph or pie diagram. And other ways to organize your data could be tables. There's plenty of different methods, and we'll look at some of them. And then finally, one of the last steps in the scientific method is the answer to your experiment. The answer may not agree with your hypothesis, but it is the conclusion to the scientific method, and it will be based on the data obtained from the experiment, whether qualitative or quantitative. And then we want to verify our results, so might as well do it all over again. So you want to retest, retrial, redo. You want to have multiple trials for an experiment. It increases the credibility of your results and your data. So it's really important to verify the results. Experiments must be retested, either by the original experimenter or others. So let's look at this example. This is a practice question, and it says a well-designed experiment manipulates only one variable. In this experiment, which is the manipulated variable? So we have mice in group A and mice in group B. They both have 1 pint of tap water. They're both in a 1 square foot cage. They both are experiencing 12 hours of sunlight. One is on a normal diet, or the mice in this group are on a normal diet, whereas the mice in this group are on a low carb diet. And we want to know what variable is being manipulated by the experimenter And in this case, it has to be what's different between group A and group B. So the answer to this practice question is diet type. So diet type is the manipulated variable. Since this is the manipulated and tested variable, is this the independent or dependent variable? If you said independent variable, you would be correct. The manipulated and tested variable is the independent variable. Let's try another question. A scientist finds a cure for cancer. She has done the experiment once. Does this prove she has the cure? Let's look at our options. Yep, she illustrated in her conclusion that she accepted her hypothesis based on her data. So since she said it was true, it's true, because she only did the experiment once, but she said it's true, and her results say it's true. Yes, her data supported her expectations. It did, but she only did the experiment once. It's the cure for cancer, so we maybe want to do multiple retrials. The third one, no, her experiment must be repeated by herself and other scientists to see if they get the same results. Yeah, that seems fair. Retesting, retrial, all that's very important to make sure your information, your data, is credible. And no, it will not be possible to cure cancer. Not necessarily true. There's plenty of research that has made treating cancer much more of a priority and more successful. We're nowhere near a universal cure, but looking at these three answers, what do you think is the best answer? If you said the third down, you'd be correct. She does not have the proof that she has the cure for cancer. Her experiment, it needs to be repeated. She's only done it once, and retesting, retrial, multiple trials by herself and others is definitely very important and would give much credibility to her cure for cancer. So let's move into another topic. Now that we've seen some examples, we've talked about the scientific method, let's talk about some of the results of scientific experiments. They can lead to scientific theories and laws. So science can be defined as knowledge about the natural world. And generally you want to tack on to that as it currently stands, because science is always changing. The scientific method, while a really great set of defined steps that includes experimentation, observation, and a key aspect of that is creating hypotheses that predict results and then produce data. Hypotheses are great again because they're testable explanations of what we see, but we know they can be proven wrong. But sometimes they're proven correct, especially after years of experimentation where we retest, retrial, and get similar, if not the same results. That makes the information that is held within a hypothesis very credible. And that's where these hypotheses can turn into scientific theories or laws. But what's the difference between them? Well, scientific theories are widely accepted. They're tested explanations for a particular phenomenon. Whereas scientific laws, they're usually something that involves a mathematical formula, it describes a certain element of nature, how it will behave, and specific conditions. Scientific laws are supported by a significant amount of evidence. They're supported by math. Again, mathematical formula are common in scientific laws. And they're a description of a natural relationship or principle that we have observed in the world. Whereas a scientific theory is not necessarily supported by mathematical terms or as large an amount of evidence. But they are something that provides an explanation. Usually an explanation of the natural world, of an observation. They usually have been thoroughly tested, but they're not something that can be proven. Some scientific theories over time have been disproven as current knowledge changed or more information became available. So scientific theories are going to be a little bit more malleable, but that doesn't make them incorrect. You have to treat scientific theories as true until more information becomes available. Treat them as the best explanation that currently exists. So a good way to think of scientific theories versus laws is scientific theory is the current explanation, whereas scientific law, a little bit more concrete, is a description supported by mathematical terms or formulae. Both do use a significant amount of evidence, but usually those with mathematical formulae are considered a little stronger. So a scientific theory is more substantial than a hypothesis because many more years of explanation, retrial, retesting, designing other experiments have come together in order not only to support an original hypothesis, but create a large body of work that has developed into a scientific theory. They usually work really well to predict the outcome of future experiments. And over time, they can be supported again and again and again and show that information in the scientific theory is solid. Or over time, more information can be made available and scientific theories can evolve. They can change, they can be revised, or they can even be rejected, depending on the amount of evidence or hypothesis testing fails that occur. Whereas scientific laws are descriptions of natural events, but they don't explain why. So theories explain, scientific laws are just saying this happens. They don't have to tell you why. So a lot of times scientific theories are a little bit more palpable. There's something that are more digestible when you're learning about biology because of that explanation that is provided. Scientific laws are a little more cut and dry. They usually occur before theories. So the description usually occurs before the explanation, meaning scientific laws often precede theories in science. So let's look at an example. Here's our example. The chromosomal theory of inheritance was developed by Gregor Mendel. So Gregor Mendel created his theory of inheritance by observing different natural phenomena and describing them. He observed the law of independent assortment, the mode of inheritance that we now know is called complete dominance. Now, the chromosomal theory of inheritance didn't exist till years after Gregor Mendel had died, but he provided a lot of the observation and data and description that produced laws that were later used to describe how we inherit genetic information from our parents. And this is still a very strong theory. It has not been changed or altered too much. But Gregor Mendel's research had to be rediscovered, and his scientific laws did precede theories in science. Of course, they were less concrete during the time he was studying that, but now we can see this natural process of scientific discovery. So let's look at a practice question. What is a scientific theory? Is it a fact that will never change? An educated guess? Or a general and reliable explanation of important natural phenomenon that has been developed through extensive and reproducible observations and experiments? Or could it be information that all scientists believe to be true beyond a reasonable doubt? If you said C, you were correct. Again, that keyword you want to look for is explanation. A scientific theory is an explanation. It even has an E in the word theory. All right, let's next look at some of the limitations of science. The processes of science do have limitations, and here are some of the things we are going to talk about. Spontaneous generation. Reedy's experiments that refute spontaneous generation. Our discovery of microorganisms, and then Pasteur's experiments. Some information about why you should study science. And then some information that talk about how some are really reluctant to let go of disproven scientific theory. So spontaneous generation and why some still cling to it. All right, so some of the limitations of science. For a hypothesis or even a scientific theory to be valid, it has to be able to be tested. Not just tested, but repeated with similar results. We want experiments that have been conducted to test a hypothesis that predicts an outcome so that we can show whether or not the hypothesis, that statement, or even the theory is false. Because it's only through experimentation, especially repeated experimentation and hypothesis testing, that we can make advances in science, that we can utilize and introduce current or new information into our understanding of science as it is today. So plenty of advances in science make everyday life better, but they're also going to make us question some past scientific theories or hypotheses or experiments. So advances may overturn previous assumptions or theories. And one example of that is spontaneous generation, because science changes take us where the evidence leads. So we have to overturn information that may have previously been thought true when new information becomes available. That's a natural process of scientific discovery and experimentation. Science looks at facts, or the current information as best it can be represented at the time. So, spontaneous generation is just a theory that emerged a long time ago, and it was the idea that life could emerge from non living matter. But that was possible. It was the idea that rats could come from straw or flies come from rotting meat. And this was commonly accepted. It wasn't laughed at, it wasn't ridiculous in the 1600s. Commonly, the part of spontaneous generation that was purported as the most true was the fact that maggots spontaneously were generated on meat. Now, they didn't realize that maggots came to the meat because it was rotting. It was just that they spontaneously, they just kind of poofed, appeared. So, spontaneous generation is what we're going to be talking about and how it has been altered. So, spontaneous generation is a theory, and it was put forth by the Greek philosopher Aristotle. Remember, it's a theory, so it was an explanation for why he observed maggots appearing on meat that was a couple days old or was rotting, and this was meat that was left out in the open. So we can guess how, for example, maggots got there nowadays. But in about 350 BC, this observation led to the hypothesis by Aristotle that living maggots were formed from non living meat. And it was given the term spontaneous generation. And it was put forward as a postulate before it became a scientific theory. And further evidence was actually garnered that kind of supported this observation. Now we know now, and we'll look at some of this, that they were not sound experiments. But even further evidence for Aristotle's law of spontaneous generation was the fact that eels have a similar smell to the slimy ooze at the bottom of rivers. So it was thought that the slimy ooze at the bottom of rivers produced eels. Not that a lot of this slimy ooze was either cyanobacteria or decaying life matter that lives at the bottom of riverbanks, or maybe even that some of the slimy ooze came off of the eels. So notice how a lot of the reasoning here, a lot of hypotheses are not sound. But it's fine that the hypothesis is not sound. What was unfortunate is at this time they didn't have enough information to conclude the hypothesis was wrong, and a scientific theory or explanation was generated, and that was spontaneous generation. So experiments, like I said, they supported the law of spontaneous generation all the way through or into the 1600s. And support for that law, spontaneous generation, continued for 1900 years. One example is biologist John Baptiste von Helmont. He put a sweaty shirt and grain in a closed wooden box, and then he would find mice in the box. After time, and about 21 days it would take, he would find mice got into the box with the grain and the sweaty shirt. Now, we could probably guess that the mouse was chewing a hole in the box and was getting in the box that way. He didn't spontaneously appear from grain and a sweaty shirt. But that's what the law of spontaneous generation said. So these were faulty experiments. They were repeated multiple times, but in the same faulty way. And so this is just showing you how a long period of scientific research seemed to support the law, so it became widely accepted. There's also Francesco Redi. He's an Italian physician. He questioned that law of spontaneous generation. Remember, the law that had been purported since Aristotle's day. But he argued that the experiment, Redi argued, that the experiment that Van Helmont had conducted could not really tell if the mice just spontaneously appeared, and it could not prove whether or not the mice gnawed into the box, or if they formed inside the box, because the box was not in an isolated place. So he decided to do that exactly. He decided that those materials needed to be isolated, and that was the key to truly testing the law. So notice how this is also simultaneously talking about valid experiments, experiments that are conducted in a way that we really truly only have one independent variable. So what Reddy did was put different types of meat in sealed jars and left them. And this meat experiment was deemed a little easier to test in isolation because you could seal the jars as opposed to making these wooden boxes mice-proof. So he was going to test Aristotle's original observation of maggots appearing on rotted meat by putting them in isolation or sealed jars and seeing whether or not maggots really appeared on this meat. And Reddy's colleagues, they argued against his findings because they said, oh, the sealed jars cut off air supply, so there's no way that maggots could have spontaneously formed because life requires oxygen. So no oxygen was not conducive to the growth or formation of life. So he redesigned it. Instead of lids, he used fine netting. Oxygen could get through, but the insects couldn't. So it kept them out. And the result of his redesigned experiment, the same as his original experiment. No maggots formed in the meat, no matter how long it was left out. So his experiments, Reddy's experiments, not only disproved the law of spontaneous generation, he illustrated the importance of not only a well-designed experiment, but testing accepted scientific theory. Because constantly information is advancing in science, and in this example, 1900 years of prior experimentation was obviously flawed. So here is Reddy and his experiments. So we have first the sealed flask, which was argued against because, again, no oxygen could get in there, so there was no way that life could live. So then we switched to the netting or the galls. Oxygen could get in, but no maggots from flies. And then, of course, with an open, non-isolated experiment, the flies can get to the meat, lay their eggs, and maggots end up in the rotting meat. But Reddy's experiments refuted spontaneous generation. And he was correct. It was a flawed theory. Next, let's look at our discovery of microorganisms. So we're going to look at Needham, his test of Reedy's findings. Let's talk about what microorganisms are first. So microorganisms are living creatures, but they're really small and we can't see them with the naked eye. So remember back when we talked about the subdisciplines of biology and we had microbiology? Microbiology would study microorganisms and they would need a microscope. But prior to 1670, the existence of these small microorganisms was not known because we didn't have microscopes that were strong enough to view them. Magnification, first of all, at first they didn't exist, but then the magnification wasn't strong enough to view them. Then came Antoine von Leeuwenhoek, and he created one of the earliest microscopes. And nowadays it would not look much like a microscope that we use. It did have low magnification, but because of this, he's known as the father of microbiology. Van Leeuwenhoek and his colleagues really clung to the idea of spontaneous generation. And he was going to prove it on the microscopic scale, thanks to his early microscope. And one of his colleagues was John Needham, and he claimed that spontaneous generation could still occur under the right conditions. So what you're reading here or looking at is that we had people who still clung to the idea of spontaneous generation, even though they were aware of Reedy's findings, of his disapproval of the theory of spontaneous generation. They've just shifted focus now to these microorganisms that have been hard to study. And they're saying that these microorganisms, these things we can't see, that's why we don't exactly know what's going on, and this is probably the reason behind Reddy's findings. He couldn't see everything that he needed to see, and spontaneous generation was still a thing. So, Needham, John Needham, along with another colleague, Spallanzani, they tested Reddy's findings. So let's first talk about John Needham. He took a flask, and in the flask, he put nutrient-rich chicken broth. This is called an infusion. And he sealed this infusion with a cork. Then he heated the chicken broth for just a short period of time. He wanted to make sure that he heated it to kill any life, because oftentimes microorganisms can't withstand a certain amount of heat. They don't like extreme conditions. And then several days later, after he had heated the infusion, he came back and looked at the chicken broth under a microscope, and he saw microorganisms. So he concluded that after several days, spontaneous generation had led to the production of microorganisms in the infusion. So he claimed, you see here, microorganisms had arisen from the nutrient broth itself. But you probably already see the flaw. The flask wasn't completely sealed because he used a porous sealant. A cork is porous, there's holes in it. Microorganisms are very small, and they can fit in the cork. Also, he may not have heated the infusion for a long enough period of time. This was early microbiology. So the flask was not completely sealed, and the bottle, therefore, was actually open to the air to a certain degree. And microorganisms do exist in the air. So next we have Lazzaro Spallanzani. He is someone who was a colleague of Reedy and Needham to an extent, and he repeated the experiment that John Needham had carried out. But to make sure that the bottles were sealed shut, he melted them shut, and then after melting them shut, heated the infusions, the chicken broth, for even longer period of time than John Needham had, to ensure that no microorganisms were left in the infusion. And then after he went to check it, after several days, no microorganisms were left in the infusion, meaning that most likely Needham's experiment was flawed. Microorganisms came from the air, got back into the infusion, not that they spontaneously generated. But still, some scientists claimed that the lack of airflow ultimately prevented the formation of life. So right now we have kind of here the microbiology version of Reddy's experiments. Let's just look at kind of what this experiment looked like. So instead of a cork, we melt the flask so it's completely cut off from the outside environment. And the heat is applied to kill microorganisms so that any that were in here before the experiment are no longer in there after the heating. And if it's not open to the external environment, microorganisms cannot get back in. If it's not sealed well, they get back in. If it's sealed, they don't. But the flaw, again, that was pointed out is most life cannot live in the absence of oxygen. So that leads us to Pasteur's experiment. Still kind of on the microbiology stage. And Louis Pasteur, he did another infusion experiment. And he stored the infusion in a flask with a really long neck. And the way that this curved neck was, it allowed air in, but not microorganisms. So think of a flask with a really long curved neck, and it's curved so that it's hard for microorganisms to make it all the way through the curve and reach the chicken broth. But oxygen, which is much lighter and moves much faster as a gas, can make it to the broth. He still heated the infusion, the chicken broth, in the flask with the curved neck, and then after the heating, no microorganisms were present. He then tipped the curved flask to allow microorganisms trapped in the curved neck into the infusion. So he took this weird looking flask, tipped it, so some of the fluid went up into the curved neck, and when it flowed back into the bottom of the flask, it had kind of wiped all the microorganisms out of the weird curved neck, and of course they were in the chicken broth. But then he repeated it, and after a period of time, instead of doing the weird flush of microorganisms into the bottom, he broke off the curved neck, let it sit, and microorganisms appeared in the infusion. So he was able to prove that even though microorganisms cannot spontaneously generate, they can, in favorable conditions, grow in a nutrient-rich environment. So their growth and development could attract more microorganisms to the environment, even after they had been killed by heat in the initial setup of the experiment. So Pasteur basically said what's happening is even though you kill the microorganisms, more come if they can get into the infusion or the chicken broth, they do. Which makes perfect sense to us now, but was something that was highly debated, especially because of the theory of spontaneous generation. So here's Pasteur's experiment. You can see the weird curved neck flask. So we have chicken broth with microorganisms in it. Heat is applied, let the flask sit. No bacteria present, and it's a more valid experiment than Spallanzano's because oxygen can get in and microorganisms cannot. So we don't have any refutable information here saying, oh, microorganisms can't live anyways because there was no oxygen. There was. Let's do the experiment again. We're going to apply heat after microorganisms were in the chicken broth. That kills them. In one situation, we're going to remove the neck and let it sit. Microorganisms are attracted to the broth because it's nutrient rich and they can live and grow there. Here, we're going to not break the neck, but tilt the flask sideways and let it sit. And because originally microorganisms were able to get into the neck, but not the broth, this washed them into the broth, and of course they appeared. And since Pasteur's experiments and tons of other experiments that supported and had similar results, the law of spontaneous generation was revealed to be flawed. So there had to be a new law, the law of biogenesis, and that's what's now widely accepted. And the law of biogenesis states that all life comes from previously existing life. So cells come from cells. Reproduction happens when cells combine to create new variabilities. But the law of biogenesis is now what is accepted, and spontaneous generation has been set aside. But it's a great story to talk about how science is ever changing and how when new information becomes available, it must be studied and looked at, and sometimes laws and theories changed. So what does the history of the law of spontaneous generation show us? And again, it just shows us science can change. Science itself is a learning process. It has limitations, but its strength is that it does have limitations, that it accepts current information and changes as new information becomes available. Advances in sciences, whether it be microbiology or population biology, they're important because they show us that laws and theories are not 100% reliable, but they are the best we have right now, and therefore should be given some credence until they are proven otherwise. So scientific laws and theories represent the best descriptions, the best explanations that science has to offer at present. And oftentimes they represent the life's work of many scientists. As we move forward, experiments are becoming more and more sound. They're having to pass more and more regulations. And just learning about experimentation does not represent the years of processing and knowledge that goes into a lot of the scientific laws and theories we know today to still be true. So let's just take a breather before we go on to our next topic and think about what we learned. We looked at what is biology. We talked about data and observations, the steps of the scientific method. And we looked at some examples on how science is still evolving. So let's go further. Let's talk more about biology. Biology, again, is the study of life. Well, what is life? Well, organisms are considered living if they share these common characteristics. So if we take these together, these characteristics, they define life. Living things are composed of cells. There are unicellular organisms and multicellular organisms, but living things are made of cells. Living things grow and develop. Humans are a great example. Living things acquire and use materials and energy. Metabolism is an example of some of the materials we use to create energy. The food you eat is converted into chemical energy. Living things maintain homeostasis, a steady state of equilibrium that we'll talk about in much more detail. Living things sense and respond to stimuli. If you've ever had a plant and you put it on a windowsill, notice that it usually bends towards the light. It's responding to that photostimulus. And then living things contain hereditary information in their DNA. So living things contain genetic material. You contain DNA that is half from your mother and half from your father. And if we went through all of these again, we could prove that you are considered a living organism. But let's first just talk about them in more basic terms. So first, all living things are composed of cells. And cells are the basic unit of living things. They're the basic unit of structure and function of living things. We will go into a chapter eventually that talks about cells and what they're made of. But the smallest unit of living things is the cell. Growth and development can increase cell size or the size of the number of cells. So growth for cells is generally them getting bigger. When they get to a certain size, though, they generally have to split. And when you get more and more cells of a certain type, and then different types of cells come together, you can get things like a tissue, and then tissues come together and become an organ. Organs become organ systems, and then organ systems make up an organism. And we are organisms. So growth and development is definitely determined by genes, the speed, how fast, how it occurs, and it ensures that offspring will develop to display many of the characteristics of the parents. Then there's energy processing. This is where we're talking about metabolism again, and it takes place within all organisms. Sometimes we can look at just one cell and look at its metabolic processes, or we can talk about metabolism in an entire organism. But all living things require energy in order to power their activities. We even have a chemical in our bodies called ATP, or adenosine triphosphate, and that's the energy molecule for human beings. Homeostasis. I said it was kind of an equilibrium. Well, it's a process, and it describes an organism's ability to maintain a pretty constant internal body condition no matter what's happening outside. A good example is your ability to maintain a relatively constant body temperature. And that is a process called thermoregulation. And when your temperature goes up, when you are running a fever, that's an imbalance in homeostasis. And imbalances in homeostasis can be really deadly. Number 5, all living things have the ability to sense and respond to stimuli. Organisms display a wide range of stimuli responses. From simple to specialized, again, that simple example I gave you was a response to light. And all living things contain DNA. We'll eventually talk about what makes up DNA. What are the parts? But we want to establish first and foremost that DNA or genetic material can be passed down from parents to offspring. So, with that being said, let's talk about cells. Let's get into what they are, and let's look at what makes them the smallest units of an organism that are considered alive. So again, we've been talking about the study of life, and cells are life. They're the basic structural and functional units of life. They are extremely small, but cells are self-contained, and they have 3 main components. They have a nucleus, which contains DNA. They have a barrier that gives them an outside and an inside, and it lets them be self-contained. And they have tiny cell machinery, called organelles, that allows them to grow, acquire energy, maintain homeostasis, reproduce, and respond to their environment, which you'll remember are the other characteristics of life. So that means a cell by itself, a single-celled organism, for example, would be considered alive. Now, most organisms are actually unicellular. We've talked about microorganisms. Those are generally unicellular or single-celled organisms. But a lot of the organisms you know of, the ones you can see that are much larger, like plants and animals, they are multicellular or many-celled organisms. And all living things can grow because that is one of the characteristics of living things. So that means cells can grow. They grow first by enlarging, then by dividing. And dividing of cells is called cell division. Cell division is just the formation of two identical cells. The parent cell produces two identical cells called daughter cells. That means we get two new identical daughter cells from one existing parent cell. Multicellular organisms also grow when their cells enlarge, divide, or develop. So cell division is vital even in our cells. Cells that are in a multicellular organism, like our cells, an organism made up of many cells, they have different functions. Because in a multicellular organism, cells can specialize. We can get immune cells, like white blood cells, that fight infection, red blood cells that carry oxygen in vascular tissue or blood vessels. There's cells in the retina that can detect light and color, that are very photosensitive, and even skin cells that are thick and protective, that are both on the outside of the body and inside of your mouth, so they even help you eat nachos. And just like all life forms must be able to grow, they also must be able to absorb energy from the surroundings and then convert it into some type of energy that can power life functions. For example, some organisms are photosynthetic, and they absorb light energy and convert it into chemical energy that they can then use to carry out life processes. So photosynthesis is one way in which organisms obtain energy. The way we obtain energy is consumption. We eat food, and that food is broken down, and what it's broken down into fuels chemical reactions that produce energy. And the process of chemical reactions obtaining and converting energy in an organism so that the production of energy can be used is metabolism. You could also say that metabolism is the sum of all the chemical reactions in the body. There's actually two parts to metabolism, and they sound similar: anabolism and catabolism. Now, all of the processes in an organism that take simple, small building blocks and put them together to produce large chemicals or large structures, those processes are called anabolic. So building processes, taking something small, taking its parts and putting them together, that's anabolism. So anabolic chemical reactions must build. They must make or build chemical bonds. Catabolism is the opposite. This is when you take big structures, large parts of the body, such as protein, carbohydrates, fat, and you break them down. These are catabolic chemical reactions that break chemical bonds. So anabolism is a building process. Catabolism is a destructive breakdown process. I like to remember that ants build mounds so they can live there. That helps me remember that anabolism is a building process. And I like to remember that cats, if left alone in a room, are very destructive. And that helps me remember that catabolism is a breakdown or degenerative process. So here's a great diagram that shows you the difference between each. Notice we have catabolism right here. We have a large product that has many bonds between it. And if you break all of these bonds, you get the smaller subunits all by themselves. So it makes sense that catabolism is the breakdown of larger molecules. In order to break down molecules, you must break bonds, and breaking those bonds releases energy. Whereas we have here anabolism. Anabolism is the synthesis of larger molecules. So we take these small parts and we make bonds between them. So anabolism is a building bond process. It puts things together. In order to put things together, you must input some energy. So catabolism releases energy, and oftentimes that same energy is used to power anabolism in another part of the body. And again, here's how you can remember it. Ants are builders, and ants and anabolic are similar. And then cats are destructive. So catabolism, think of it as catastrophic or degenerative. And so they generally break down complex substances into simpler ones with a release of energy. So if you've ever heard of the 2 a.m. zoomies for cats, that's where they're releasing a lot of energy in order, or putting in a lot of energy into their destruction in order to break down substances into simpler ones. And that leads us into another characteristic of life, and that would be metabolism and energy. So we're still kind of playing off of the other and just looking at it in kind of more detail, because we're eventually going to be looking at kind of photosensitivity and how different organisms respond to sensory stimuli. So let's talk more about metabolism and energy and how that is going to transition into sensory stimuli. So light energy from the sun is the main source of energy for all living organisms, and organisms that can perform photosynthesis convert this light energy into chemical energy, which is essentially food for themselves, and that photosynthesis process is usually used by green plants. Plants that are photosynthetic usually have types of cells that have certain organelles or tiny machinery in them, and they're really amazing because they can take light energy and produce their own food. We unfortunately cannot do that. Humans are not autotrophs. We are not self-feeders. Instead, we're called other feeders. We're considered heterotrophs. And there's different types of heterotrophs. Of course, there are different types of plants that can eat organisms as well, so they're not purely autotrophs. But for the most part, when we're talking about heterotrophs, we're talking about animals. Organisms that only eat plants would be herbivores. Organisms that only eat organisms other than plants are carnivores. And then humans, we're omnivores. We eat both plants and other organisms. But herbivores, carnivores, and omnivores, they are all heterotrophs. They get their energy by eating others, whether it's the autotrophs themselves or other heterotrophs, and they cannot perform photosynthesis. So these are organisms that depend on other organisms for their food, and that is where we want to make the big difference. Organisms that eat other organisms are other feeders, heterotrophs, and autotrophs are generally only plants, some cyanobacteria, that can perform photosynthesis. These processes of metabolism and the creation of energy do play into homeostasis, which is the maintenance of stable internal conditions no matter what's happening outside your body. Birds and mammals, they're endotherms, whereas reptiles are ectotherms. What that means is that birds and mammals are organisms that are warmed internally by metabolic processes in their body that generate heat. Whereas reptiles, being ectotherms, they have to get their heat from an external environment. So oftentimes you may see, for example, a lizard basking on a rock, especially when it's warm out. It's using the heat from its external environment to help maintain its body temperature within a particular range. And it's that within a particular range statement that has to do with homeostasis. Because homeostasis is not one single particular measurement. It's not having a blood glucose level of 340. It's not having an internal body temperature of 37 degrees Celsius. It's actually maintaining a particular range of characteristics. So blood sugar would be within a range, body temperature would be within a range, but we are really used to kind of one particular measurement being reported or looked for. But just keep in mind that homeostasis has to do with the maintenance of stable internal conditions within a normal range, not singular numerical number. So let's look at a mechanism of homeostasis. Our body, for example, uses negative feedback systems. And negative feedback systems are pretty simple. For example, if our body temperature increases, processes in our body are going to work to cool it back down. So a negative feedback loop is like what you see here in this slide. So here we have a person right in the middle. They're kind of within normal body temperature range, which for humans is 37 degrees Celsius. And right now it's hot, or if it was hot outside, body temperature would increase. And as body temperature increases, there are signals in the human body that activate cooling processes, such as the release of sweat from sweat glands. Sweat from sweat glands has a lot of water in it. And when water absorbs heat on your skin and eventually evaporates, it takes that heat with it. And since the sweat evaporates and takes heat with it, it cools the body and body temperature decreases. What about in December here in Georgia? And you're standing outside for even a short period of time, your body temperature might decrease. And that could cause shivering. Shivering is actually the contraction, very quickly, of skeletal muscles. And that metabolic action of muscle contraction produces heat as a byproduct. And that heat produced by muscle contraction actually raises your body temperature. So you return back to normal. So notice that with a negative feedback loop, whatever the original stimulus is, ends up being reversed. So the original stimulus here, when you got hot, was body temperature increases, and the end of the negative feedback loop was body temperature decreases. Here on the left, body temperature decreased because you were cold, and then the end product of the negative feedback loop was body temperature increases. So undoing the original stimulus is the goal of negative feedback loop, and this is how most homeostasis is maintained in humans. A positive feedback loop, the opposite of a negative feedback loop, can be very damaging, and it usually only happens in illnesses or homeostatic imbalances that are not maintaining a normal range of homeostasis in the human body or in other organisms. And this finally leads us to sensing and responding to stimuli. And organisms obviously can perform metabolism, create their own energy, monitor their boundaries. They can also perform homeostasis. But probably the one you're most used to is sensing and responding to stimuli. You do it every day because an organism's ability to sense changes in their environment is just as important as its ability to respond. You're constantly taking in information through your eyes, your ears, mechanoreceptors, in your fingers, or along your back. So it's very typical that what defines a lot of life for many organisms, not just humans, is their ability to take in information, perform observations, look at their surroundings, and then process that and respond. And we see this in not only humans, but even autotrophs like plants. But living organisms are all equipped with some method of receiving information about their surroundings, and this method generally involves or is accomplished with receptors. A receptor is a structure that allows for processing of sensory information. Plenty of sensory organisms in animals detect and respond to external stimuli. Light, sound, and chemicals are probably the ones you're the most used to. If, for example, you like to go to Chick-fil-A and you like Chick-fil-A because they put pickles on their sandwiches, then you might like, for example, sodium as a chemical because pickles usually have a lot of salt. And that would be a chemical that the receptors in your mouth are processing. Those receptors would be your taste buds. But there's also plenty of internal stimuli that we don't necessarily have to think about on a daily basis. And internal stimuli in animals are perceived by things like stretch and temperature, and really extremes like pain. Chemical receptors, especially, are good at picking up on pain stimuli. Like if you eat too much and your stomach is very full, that stretching becomes extreme and leads to pain or an upset stomach. Sensing and responding to stimuli is also an adaptation that organisms must have in order to be able to survive. For example, hearing whether or not a predator is approaching, or seeing if a predator is approaching, or noticing when it's nighttime, and knowing whether or not you're a prey animal or a predator that likes to hunt at night or needs to hide at night. So animals, plants, fungi, and even bacteria can respond to stimuli. So here is the basic effector response, and it all begins with sensing a stimulus. So the stimulus is sensed by a receptor. Here, you are seeing water, and that's being picked up by photoreceptors in the retina of your eye. That information has to go from the photoreceptors in the retina of your eye to your brain. They do that via a sensory neuron. Then your brain thinks about this information, it processes it, and it sends that information to your muscles or to an effector neuron. This effector neuron goes to an effector, and an effector has an effect. And usually the effect in the human body is muscle contraction or secretion, like a gland secreting sweat. But here, the effector motor neuron is going to cause movement of the skeletal muscle in your arm because you saw water, when your brain processed it, realized you were thirsty, so it told your arm muscles, realized you were thirsty, so it told your arm muscles to contract and to pick up the water so you can drink it. So this is a simplified version of a complicated process that involves responding to stimuli and an effect happening because of that. Another example that's a little simpler is light sensitivity. So this is a response to stimuli from a plant. So plants are light sensitive, and that's called phototropism, and it's the orientation of a plant or other organism in response to light, either toward or away from the light. If it's toward, it's positive phototropism. If it's away, it's negative phototropism. And then finally, to one of our last characteristics of life, DNA and reproduction. All life forms reproduce. There's asexual and sexual reproduction. Asexual reproduction is the process by which a single organism produces genetically identical offspring, and this offspring receives all DNA from one parent. So asexual reproduction is similar to cellular division. A lot of unicellular or one-cell organisms perform asexual reproduction. Then there's sexual reproduction, where you have two parents produce a genetically different offspring. So in asexual reproduction, the offspring is a clone of the parent. In sexual reproduction, the offspring is genetically different from the parents. So the offspring would receive a combination of DNA from their two parents. And reproduction, regardless if it's asexual or sexual, involves the concept of inheritance, which is the passing down of traits to offspring, or the process by which physical and biological characteristics are transmitted, passed down from the parent to sometimes the daughter cells, sometimes the variant or genetically different offspring. And during this process of inheritance, mutations can occur and cause offspring to inherit traits that are different from their parents. In asexual reproduction, if a mutation occurs, it can cause what should be a clone to be slightly different from the parent. Because mutations are really abrupt and marked changes in the DNA of an organism, especially compared to that of its parents. And that's important because DNA is the blueprint for how that organism should run. It's the set of instructions. DNA contained in the chromosomes that are in the nucleus of a cell contain chemicals that make up life and how it should be processed, produced, or work, especially in a multicellular organism. So let's try some practice questions that relate to the characteristics of living things. Here's our first one. Our body works to keep our temperature at about 98.6 degrees Fahrenheit, which is about 37 degrees Celsius. This is an example of which characteristic of living things. A, living things are organized. B, living things respond to stimuli. C, living things maintain homeostasis. Or D, living things need energy. If you said C, then you would be correct. Living things do maintain homeostasis, like maintaining body temperature. A great example is maintaining thermoregulation, maintaining a body temperature of 98.6 degrees Fahrenheit. Let's look at the next one. If you put a plant in your window and the stem will lean in the direction of the sun, this is an example of which characteristic of life? The first one, living things are organized. Second one, living things respond to stimuli. Third, living things maintain homeostasis. Or the fourth, living things need energy. If you said the second one, or B, living things respond to stimuli, you would be correct. Remember, that was phototropism. Next, let's look at the tools of biology. Pretty much a common measurement system. If you've taken physical science, you've learned a lot about measurement, especially the metric system, which is commonly used in science, like physical science, physics, chemistry, and of course, biology. In order to study living things, we require plenty of tools, procedures, equipment. Scientists use things like a balance, a microscope, telescopes, computers, robots, in order to see things that are small, or to measure the mass of a specimen, or see something far away, or even to work with small things like data about DNA. But studying living things does require a standard measurement system, so that studying biology here can be translated or sent to another location. And while here in the US, we tend to use the imperial system, the metric system is the system of science. The metric system uses units that are scaled to multiples of 10, and another name for this metric system is the International System of Units, or SI. And most scientists are going to use this metric system, especially when measuring lengths, volumes, masses, and temperatures. Other tools of biology, tables and graphs. Very important, especially when you're writing down all of your data that you want to communicate to other scientists. Once measurements are taken and data is collected, scientists need to organize, analyze, and interpret the information they've gathered. Charts and graphs are also useful for finding relationships or trends between variables in experiments. And that leads us to one of the most important tools of biology, modern-day microscopes. They're able to magnify images of structures that are too small to see with the naked eye. And they're also able to produce large images while still remaining sharply focused. We'll be using light microscopes in this class, and they're really the most commonly used type of microscope. They're oftentimes called compound light microscopes because they compound multiple lenses in order to produce a clear image, and magnifications of 400 or 1,000 times, especially when using oil immersion, are possible. Compound light microscopes use a combination of two lenses to form an image. The specimen would be observed, but it must be thin enough for light to pass through so that the lenses can produce the image. Now, light microscopes are what we'll focus on and look at the most in this class, but it'll be interesting to look at even fancier microscopes like the electron microscope, which in the 1950s were developed by biologists. Electron microscopes use lenses made of electromagnets, and they can gather and focus a beam of electrons, and the specimen would be placed in a vacuum in order to ensure the beam of electrons travels directly to the specimen and doesn't escape. You would want them to be traveling right at the specimen so that you would get a very clear image. There's different types of electron microscopes, and they can form images of objects 1,000 times smaller than objects magnified with light microscopes. The two types of electron microscopes are the TEM and the SEM. Transmission electron microscope is the TEM, and it's a microscope that transmits a beam of electrons through a thinly sliced specimen, and it's to look at the internal structures. So transmission electron microscopes look at the internal structures of really small objects, whereas scanning electron microscopes are looking at the outside. They're looking at the surface of a specimen. So it would be a microscope that passes a beam of electrons over the surface of a specimen because it's focused on the external topography or outside surface of that particular specimen. Electron microscopes, unfortunately, can only be used to view dead specimens, whereas light microscopes can be used to view living and dead specimens. So there are benefits and negatives to both. So let's look at experiment 1.1, Introduction to the Microscope. We'll look at the parts of the microscope and more. So in general, the rules for using a compound light microscope is to begin focusing with the shortest objective lens, called the 4x or 4 times magnification lens, in place. And you want to move it from low to higher magnification as needed. So you always start with the lowest power objective lens. And when you use the lowest power objective lens, you would use the coarse adjustment knob. So the 4x or 4 times magnification lens is also known as the scanning lens, and you always use it with the coarse adjustment knob. And you only use the coarse adjustment knob with the shortest or 4x objective lens. And you'd want to store a microscope properly when finished, which means you want to make sure it looks the same as it did when you got it. And you always want to use 2 hands to carry it. So here is a compound light microscope. Up here are some lenses, and down here are 3 more. These are the objective lenses. The ocular lenses are the first pair. Notice they kind of look like binoculars. And with most ocular lenses for a compound light microscope, they are going to be at a power of 10x, or 10 times magnification. Then you have the head. The head supports the ocular and objective lenses. Then you have the arm, which connects the head to the base. Power switches and light controls tend to be on the arm or sometimes the base. And a power switch would turn the microscope on and off. You have also a light control, which turns on and off the light, but it also regulates how bright that light is. The light is right here, and it's under this flat platform called a stage. So the light is generally called a substage light. Let's look at some more parts of the microscope. The stage is this flat platform. This is what your microscope slide would go on. The mechanical stage is all of this metal here, and that's about where you would put your slide. Here's a stage clip, but that's all a part of the mechanical stage. It holds the slide in place and allows for precise movement of the specimen. Then you have the coarse adjustment knob. Notice that we have a bigger oval here and then a smaller oval within it. On the other side, this would be even more pronounced, but the bigger oval here would allow the stage to move up and down, and that's a coarse or rough movement. So it'd be used for initial focusing, and it moves the stage up and down. Then the fine adjustment knob is the smaller knob on the side, and it allows for fine focusing at higher magnifications. So you would always start with the really short objective lens and the coarse adjustment knob, and then as you move up to higher power, you would switch to the fine adjustment knob only. Now this moves the stage up and down slightly, but you're not going to see it, because really what it's moving is how close the lenses of the objective lenses are to the stage. The base again supports the arm and most of the microscope components. It is connected to the head via the arm. The rotating nose piece is right here, and you can turn it, and it carries the objective lenses, and you would rotate it to change objective lenses in use. The objective lens in use is the one facing the stage. And the objective lenses, which are right here, they would magnify the specimen, and each lens has a different magnification. Let's look at some other parts you might not have seen. This is a different view of that microscope. You have the condenser, which is right here. It concentrates light on the specimen. The lens for that would be right at the top. Iris diaphragm lever is this lever, and it would open and close the condenser lens, kind of like your eyelid opens and closes the ability of light to get in through your pupil, because it's regulating the amount of light passing through the condenser. It's using a name similar to the colored part of your eye, the iris. The substage light provides a light source. We pointed that out in another view, but it's under the stage, so substage. Sub means below. And mechanical stage control, so that metal that was on the top of the stage can be moved around using these knobs right here, and that's going to move that metal on the stage called the mechanical stage. But since it's moving it, they're the mechanical stage control, sometimes control knobs. We also want to just talk briefly about some terms related to microscope use. So you can figure out the total magnification of the microscope that you're using, because you would multiply the power of the ocular lenses times the power of the objective lenses. So if the power of the ocular lenses is 10x, and you're using the scanning objective 4x lens, you would multiply 10 times 4 to get a total magnification of 40x. And that would be the total magnification used at that point in time to look at your specimen. There's also the field of view. Field of view is the maximum area visible when looking through the eyepiece. So if you look through the ocular lenses, you would see a circle, and depending on the magnification you're using, you would see a specimen that is smaller or bigger. As you increase magnification, the specimen gets bigger, but the amount you can see around it, your field of view, actually decreases. Working distance is the distance or space between the stage, or your specimen, if it's on the stage, and the bottom of the objective lens. And since the objective lenses get longer as you go up in magnification, as you increase magnification, the space between the objective lenses and the stage of your specimen decrease. Here's some more on total magnification. Most of the microscopes we'll use in class have 3 objective lenses. A scanning lens that has a 4x or 4 times magnification, a low power lens that has a 10x or 10 times magnification, and a high power lens that has a 40x or 40 times magnification. So we want to assume that the ocular lenses for all of our microscopes is just 10x. For most compound light microscopes used in a scientific classroom, the ocular lenses are going to have a magnification of 10x. And when you look at a microscope, we'll look at some in class, they actually have written on them what the magnification is. So the scanning lens will have a 4x, low power 10, high power 40x. And to calculate total magnification, you would just need to know which objective lens is in use. So if you're using scanning, it's 4 times 10, low power 10 times 10, high power 40 times 10, which would give you total magnifications, depending on which objective lens you're using, of 40x, 100x, and 400x. So you want to practice calculating total magnification of a microscope by using the above information, and we will do that in class. Let's talk about some of those terms, such as working distance, field of view, things like that. They have relationships with magnification. So as you increase total magnification, field of view decreases, working distance decreases. So that means that magnification, field of view, and working distance, they have inverse relationships. And then the amount of light needed may increase if you increase your total magnification in order to see something clearer, to see the details. So total magnification and the amount of light needed, they have direct relationships. But think about it. If your image is not centered, you may lose it altogether at a higher magnification as your field of view decreases. As you use longer and longer objective lenses, which are higher magnifications, you have less space between the objective lens bottom and the stage, so that's your working distance decreasing. And again, the amount of light needed may increase if you increase magnification in order to see all the tiny, tiny details. And that is it for our first chapter in biology. And when I see you on Friday, we will begin looking at microscopes in even more detail. Bring any questions you have so that I can answer them.